The primary role of the lung is to bring atmospheric air and pulmonary capillary blood into close enough proximity to exchange oxygen and carbon dioxide, and then transfer that blood to the left atrium of the heart and then to the rest of the body. The ventilation of the alveoli and the perfusion of the alveoli, they need to be matched to allow for adequate gas diffusion across the alveolar membrane. In this lecture, we'll be reviewing oxygen and carbon dioxide transport and how it moves throughout the body. Pulmonary medicine thrives on understanding the partial pressures of oxygen and carbon dioxide and how they change. And so we're going to be spending some time getting to know how these gases move across the alveoli, hemoglobin across peripheral tissues, and we'll cover what these numbers mean in health and during disease. So to best understand gas exchange between the air and the lungs, let's look at a couple of different gases here. So I'm going to write down a few. We've got oxygen, nitrogen, carbon dioxide, we've got water vapor, and we've even got argon. How about that? So what I'm going to do is with each one of these gases, I am going to put them into their own individual container where they're the only gas inside of this. Next, I'm going to put them all at sea level and the same temperature as well. And so if I put a little barometer inside each one of these glass containers holding each of these individual gases, what we would see is we would see that each one of these gases exerts its own pressure inside of these little glass containers, and they're all individual. So this pressure is measured in millimeters of mercury. And let's say if we wanted to put all of these gases into one single container, again with oxygen, nitrogen, CO2, argon, water, etc., we would find that each of the individual pressures of each of these gases would add up in here. So each one of these partial pressures of the gases added up equals the sum total of all of the gases' pressure combined here. This is what we call Dalton's law, as seen in this equation here. So our atmosphere, as seen in this little container here, is a mixture of a number of gases. And like we've demonstrated already, it's primarily oxygen, nitrogen, with very, very minute small amounts of carbon dioxide, vaporized H2O, and argon. And at sea level, our atmosphere's total pressure is 760 millimeters of mercury. This is a total pressure resulting from all of the individual partial pressures of the composition of dry air. So all of these are going to have all of their own pressures. Let's have a better look at what this means in the next slide. So if I took two samples of atmosphere, one is at sea level and one is at the top of Mount Everest, what we would see is that the atmospheric pressure, as we already talked about at sea level, is 760 millimeters of mercury. And at the top of Mount Everest, it's less than half, so it's less than 300 millimeters of mercury, because there is less gas molecules up there. However, what we would see is that if we took both samples we would see that the fraction or the percentage of each of the amount of each individual gas is always going to be the same. So what this is called the fraction of inspired gas, or here the fraction of inspired oxygen, is always about 21%. And same thing for both canisters of air. We would also find that the fraction of inspired nitrogen, for example, would be 78%, and the fraction of inspired CO2 would be less than 1%. So that said, when we go back to sea level, which is more compatible with human life and where most of us live, the main thing that we want to know in pulmonary medicine, outside of the fraction of inspired gas, is we want to know the partial pressure of the gas, because this is essentially our pathway into understanding how gases move from the atmosphere into the blood. So the partial pressure of oxygen is going to be 160 millimeters of mercury, and the partial pressure of CO2 is going to be 0.23 millimeters of mercury. These partial pressures are measured in pulmonary medicine because they allow us to diagnose gas exchange issues and, more importantly, acid-base disturbances. Let's keep going. When we breathe air from outside of our body, it enters the lungs via the trachea. It enters the bronchi, it enters the segmental bronchi, the terminal bronchioles, and eventually it enters the alveoli. So as air enters the bronchial tree, it is humidified, and you can see this change here. Increasing water vapor content, it mixes with residual air left over from the previous breath, and gas is also being exchanged actively in the alveoli. So for these reasons, we can measure and we can track the differences from inspired air to what's actually found in the alveoli. So can you see this difference here? So what we begin to see is we begin to see how the partial pressures change, and these numbers are very important to know. These numbers are very important because the alveoli has a very stable microenvironment where the partial pressures really don't change that much, even in response to rest or exertion or rock climbing or sometimes even disease that's non-pulmonary. So these are good steady state numbers, especially this 40 and this about, we can call this 100, we can round down. These are important numbers to know that talk about the steady state of the alveoli. So there are 5 factors that influence gas movement through the alveoli, through the tissue, and then into the blood. So let's begin at ground zero. Let's begin with an alveoli, and let's begin with an alveolus, and here we have a pulmonary capillary. So we can see the single file line of red blood cells moving through. So inside here we have alveolar microenvironment or alveolar air, and inside here we have both oxygen and CO2, and then also over here we have oxygen and CO2 inside of the blood. Now watch carefully how I annotate this here. So for alveolar, when we're talking about the partial pressure of oxygen inside the alveoli, I'm going to annotate it partial pressure, P capital A for alveolar, and then the gas. So we have partial pressure of alveolar oxygen is roughly 100 millimeters of mercury, and the partial pressure of alveolar CO2 is roughly 40 millimeters of mercury. Down here, the partial pressure, and note this, of arterial oxygen is 40 millimeters of mercury, and the partial pressure of arterial CO2 is 45 millimeters of mercury. And again, these are the partial pressures inside each environment. So inside the alveolus and then inside the blood and plasma. Okay, so the first rule in gas movement is that gases move down their pressure gradients from high to low. Here we see that the differential of partial pressure of oxygen is about 60 millimeters of mercury between here and here, and about 5 millimeters of mercury with the carbon dioxide. This means that both will passively try to equalize themselves. The drive for the diffusion is the partial pressure of gas. The second factor in gas diffusion is how easily the gas dissolves in water, or rather the solubility of gas. So carbon dioxide, for example, is much more soluble in water than oxygen is. On the other hand, oxygen has a much steeper partial pressure gradient. Again, it's about a 60 millimeter of mercury difference. So in the end, carbon dioxide and oxygen exchange rates are about the same, and they occur in equal amounts. So third, membrane thickness is going to affect how well gas diffuses into the blood. So the baseline thickness of an alveolus is about 0.5 micrometers at best, and it really doesn't pose much of an obstacle for gases to diffuse in and out. But when it thickens, this could be due to chronic inflammation. This could also be due to perhaps mucus or pulmonary edema. This is going to pose more of a challenge. And what we'll see is that gas exchange is often slowed or even inhibited because there is a much larger distance to cross. Another factor affecting gas exchange is the membrane surface area. I've said in the past that the total lung respiratory zone is about equivalent to that of a tennis court. Well, in diseases like COPD, it can be reduced to that of something like a table tennis court. Lungs are heavily reliant on simply the massive scale of diffusion, and in things like emphysema, the alveoli walls can be scarred or destroyed, reducing the surface area. So the last concept is ventilation and perfusion is absolutely essential for gas exchange. So here, the alveolar capillary unit needs two working components. It needs the alveolus for ventilation, and it needs a good pulmonary capillary flow for perfusion, which we annotate as Q. So when we have good ventilation, we have good healthy airflow, and when we have good perfusion, we have a good blood flow available for gas pickup and drop off. Now, when these two are in good working order, we call this matched VQ. On the spectrums, you'll be able to see how this can fall apart. So if we have terrible ventilation over here, but we have good or decent perfusion, this is a mismatch. And on the other side, we can also have really good ventilation, but we might not be able to get blood to pick it up. So this is also a mismatch. So a mismatched ventilation perfusion ratio essentially means that one or both of these components aren't working. Here's a quick summary of the 5 factors affecting alveolar gas exchange. Gases moving down pressure gradients, gas solubility, alveolar thickness, alveolar surface area, and then of course VQ matching. So let's put all of these numbers together. Beginning at the top, inspired air has a PO2 of about 160 millimeters of mercury, and the PCO2 for all intents and purposes is 0. As it's inhaled and touching on the previous slides, we see that PO2 is now about 100 and PCO2 is about 40 millimeters of mercury. So here, gas moves down its gradient, oxygen moves into the capillary, and CO2 diffuses into the alveoli. And moving further down, blood is compartmentalized mainly to the large and medium-sized arteries. It's not in the peripheral tissues yet. So we'll say in systemic arteries, the PaO2 is 100 millimeters of mercury, and the PaCO2 is about 40 millimeters of mercury. Once it's arrived in the peripheral tissues, like a muscle, the pressure gradient facilitates gas transfer. The difference from arterial circulation to tissues is about 60 millimeters of mercury of oxygen. And blood also takes up carbon dioxide as a waste product from the tissues. We can see that the cellular partial pressures are 40 millimeters of mercury of oxygen and 46 here, roughly 45, just for simplicity's sake. PCO2 in tissues is about 46. And once post-circulation, that PO2 to PCO2 ratio of 40 to 45 remains. And it really remains this way, generally speaking, all the way until it arrives into the right atria and then the right ventricle and gets ejected into the pulmonary capillaries. And when it arrives here, it undergoes that same diffusion process and it starts all over again. Understanding how oxygen and carbon dioxide are transferred in the blood is extremely important in pulmonary medicine. As we know that oxygen is primarily bound to hemoglobin, but there is an unbound portion that is dissolved in plasma comprising of about 1.5%. Carbon dioxide transport is carried through the blood in roughly 3 separate mechanisms. The primary mechanism is on the bicarbonate ion. However, it's also able to hitch a ride on hemoglobin and be dissolved in the blood plasma itself. So these are 3 different ways to think of oxygen when we try and answer this question, how much oxygen is in the blood? So we'll start with number 1, the partial pressure of oxygen in arterial blood. So the PaO2 is the driving force for oxygen to enter the blood from the alveolar capillary system. And what this does is it reflects the free oxygen dissolved in plasma and not the oxygen that is bound to hemoglobin. For that, we're going to need the next one, the hemoglobin saturation. The SaO2, or the hemoglobin saturation, is the total percent binding sites on arterial hemoglobin saturated with oxygen. We already know that binding sites for oxygen are located in the heme groups on hemoglobin, and there are 4 heme sites per hemoglobin molecule. Heme sites occupied by oxygen molecules are said to be saturated with oxygen. So the percentage of all of the available heme sites saturated with oxygen is the hemoglobin oxygen saturation, or the SaO2. So note that this is really oxygen that is bound. This is the oxygen that is unbound. And you can see that in order to get both of these, we turn to number 3, or otherwise called the oxygen content. So the oxygen content is measured in milliliters of oxygen per deciliter, and it's the amount of bound and unbound oxygen molecules per unit of blood. The CaO2 is the only value that incorporates the hemoglobin content. Unlike either the PaO2 or the SaO2, the oxygen content directly reflects the total number of oxygen molecules in arterial blood, both bound and unbound to hemoglobin. And the CaO2 depends on the hemoglobin content and is directly related to the hemoglobin content. So the normal CaO2 range is from 16 to 22 millimeters of oxygen per deciliter. Oxygen is primarily transported by being chemically bound to hemoglobin in the erythrocytes. And the hemoglobin rapidly and reversibly binds to oxygen. And for hemoglobin to become fully loaded with oxygen molecules, it takes about 0.01 seconds. The reversibility of the reaction allows oxygen to both be loaded and unloaded into metabolically active tissue. That's very important. So what is hemoglobin? Hemoglobin is a polypeptide made up of 4 heme units with 2 alpha and 2 beta polypeptide chains, and sometimes there can be some genetic variability in these polypeptide chains, leading to things like thalassemia, which you're familiar with in your heme unit, which is way beyond the scope of this lecture for now. But in essence, each subunit contains 1 iron molecule, and each iron molecule is capable of binding and releasing 1 oxygen molecule, which translates to 2 oxygen atoms. So in its oxygenated state, we call this oxyhemoglobin. In its deoxygenated state, we call this deoxyhemoglobin. And the hemoglobin increases the oxygen carrying capacity of blood about 100-fold over plasma. Looking at these two here, and here we've got hemoglobin saturation, and here we've got the oxygen content. Now, in relation to the last slide that we just talked about, hemoglobin saturation, or the SaO2, it describes the amount of oxygen that is bound to hemoglobin in arterial blood. So this term saturation kind of relates a hemoglobin to something like a sponge that becomes saturated with oxygen. Its measurement is given as a percentage. The oxygen content is both the bound plus the unbound content of oxygen, and this is in milliliters of oxygen per deciliter. So the kind of interesting bit about this here is that the SaO2, the stand-in for the hemoglobin saturation, which we'll talk about in another lecture, is going to be the SpO2, or the peripheral oxygen saturation, or this is called the pulse oximeter. Hemoglobin doesn't absorb oxygen evenly or in a linear fashion. So this image here demonstrates 2 different molecular configurations of hemoglobin. One, deoxygenated hemoglobin, or hemoglobin with no oxygen attached, and oxyhemoglobin, quite the opposite, a fully populated hemoglobin molecule, which we can see here. So in its deoxygenated state, the bonds between the globin units are tight, and I'll use red for this. And so these bonds, the molecule overall is very unfriendly to oxygen molecules that are interested in bonding. So this configuration we call is a tense state, and we say that there is low affinity. And affinity, as I've described it here, it's a chemical attraction force that causes molecules to want to combine. When the molecule of oxygen eventually does bind with hemoglobin, what it does is it changes the entire molecular structure of the hemoglobin molecule. It's not the same as it was previously. And what this does is 1 oxygen binding to hemoglobin now opens up and exposes more oxygen binding sites. So we say that the affinity increases. In this image down here, we have a T, or a tense state, where we have a tight hemoglobin configuration. And as 1 oxygen molecule binds, we can see that these now turn purple. And the purple, as we can see, these now have an open configuration. So the affinity for this, and we'll say this unit, are now open for business. They are more interested in bonding with oxygen. So when the next oxygen, this is number 1, this is number 2, when the next oxygen molecule binds, we can see that now we have 2 more friendly units open up. And as number 3 shows up, we have a fourth. And as number 4 attaches, we have a fully, what's called an R state or a relaxed state. So in summary, each consecutive oxygen attachment actually makes it easier for the next oxygen to attach. So this is not a linear progression of oxygen attachment. This is more of a sinusoidal curve. And it actually has a name. It's called the hemoglobin oxygen dissociation curve. Let's have a look at it. The oxygen hemoglobin dissociation curve illustrates hemoglobin's affinity to oxygen. And I'm going to write this out here. So affinity between hemoglobin to oxygen. So in other words, the graph shows the relationship that hemoglobin has with oxygen when there's varying amounts of oxygen or varying partial pressures of oxygen. So to review the x and the y-axes, the x-axis is the partial pressure of oxygen from 0 to 100 millimeters of mercury. And then we'll give this a different color. The percent saturation of hemoglobin is 0 to 100% saturation. So something to note here, given the chemical properties of hemoglobin that we discussed in the previous slide, hemoglobin doesn't bond to oxygen in a linear fashion like such. It doesn't do this. I should say the affinity is not linear. It actually has more of an S-curve to it, which means, and I'll generalize this, that there are two main sections of affinity, or the two main characteristics of affinity. The first I would say would start at the T or the tense state and go up to about 75% saturation of hemoglobin, where there's a relative low affinity for bonding. And then the next is going to be from 75% to 100% of hemoglobin saturation, where there's a relative high affinity for bonding. And I think to illustrate this, we can also use the numbers down here, which I forgot to connect. So I'm going to start at the top right of the graph at 100 millimeters of mercury, right here, which is approximately that to alveolar air, which I will write here. Note that from about 60 millimeters of mercury on up, we have what's called a plateaued region. And I'm going to box it off here. Plateau, excuse me. Here the affinity of hemoglobin to oxygen is really high, meaning that the bonds, this bond right here, is strong and it's difficult for oxygen to break free. So at 100 millimeters of mercury of oxygen, hemoglobin is actually only 97% saturated. So it really, in this area, it takes a lot of oxygen after 60 millimeters of mercury to maximize hemoglobin saturation. So let's move down to 40 millimeters of mercury, which is a long way away from 100. So at 40 millimeters of mercury, hemoglobin is saturated to about 75%. And this number is important because 40 millimeters of mercury represents the partial pressure of oxygen in most resting peripheral tissues. And this area here, which I'm generally going to block off, like so, is called the steep section, or the steep region. And this is where affinity becomes remarkably less as oxygen partial pressure decreases in the tissues. So this means that the less oxygen that's available, moving to the left, the easier it is for oxygen to unbind to hemoglobin as that affinity reduces. So plateau region overall is going to be a high affinity region where there is a stable bond. And the steep region has a reducing affinity the further deep into oxygen-starved tissue that you go, and this is a weaker or an unstable bond. And that's why we get this S-curve. It's because it's not linear. So in summary, if you have high, or the higher the PaO2 goes, the higher the affinity goes between the bonds, between hemoglobin and oxygen. And the lower arterial PaO2 goes, the lower the affinity goes as well, which leads to more oxygen available. So this is how hemoglobin innately understands and reacts to tissues that need more oxygen. They can deliver oxygen to tissues where it's needed, and they can hold on to oxygen in areas of the body that don't need it. So to reiterate, the hemoglobin-oxygen dissociation curve illustrates affinity between these two molecules. And really, when we're talking about affinity, we're talking about a number of things, but the one that I'd like to highlight here is bonding between the two. And whether these bonds are either stable or unstable. And when they're unstable, they dissociate. So hemoglobin and oxygen, and again, we can kind of exchange, we can talk about affinity with a number of different things. It doesn't have to just be between these two. We can talk about the affinity of hemoglobin to carbon monoxide. We can also talk about the affinity of fetal hemoglobin instead of adult hemoglobin, but let's stick with what we got right now. So now that we understand the big picture of this dissociation curve as being about chemical bonding or affinity, let's add in a couple of other variables to see how it might relate to tissues that might need more or less oxygen. So one thing to note here before I get going is that hemoglobin's molecular structure can be influenced by its environment. And I'm going to write environment will change, that's delta, will change hemoglobin structure. And the environment I'd like to talk about first is a muscle that is running a marathon. Here the temperature will be a little bit higher, the partial pressure of CO2 will be elevated, which will reduce the pH or increase the acidity. There will be more of a molecule called 2,3-DPH, which is found in the red blood cells. So this new environment here that a red blood cell is plunging into when it goes into muscle that is running a marathon actively influences the bonds or the affinity for oxygen, and they weaken this bond. And what this does is this moves the baseline dissociation curve to the right. So when the affinity is weakened, it requires more partial pressure of oxygen to saturate the hemoglobin molecule. And on the flip side, it's easier for oxygen to escape. So here we see the resting state of this runner is it requires about 26 millimeters of mercury to saturate the hemoglobin to about 50%. And in a right shift, which I'll do in red, it looks like that those oxygen requirements have actually increased to saturate the hemoglobin at 50%, which means there is more oxygen that is dissociating from the hemoglobin to active muscle. So in summary, a right shift means that it is harder for oxygen to bond, and it is easier for oxygen to be released into the tissues. So what about a left shift? Well, let's talk about a muscle that is cooled off or is resting. This is our second microenvironment. Really, in any muscle or in any portion of the body that has reduced temperature, that has reduced partial pressure of CO2, reduced 2,3-DPH, or kind of a more alkaline environment, we see that hemoglobin tends to hold on to oxygen more, and this produces a left shift. So here, overall, it's the affinity is stronger, and the curve is more steep, which indicates that oxygen is being held or bonded by oxygen better, and it's harder to release. So what I'm going to write here is that it's hard to release. And we can actually see this in action because it looks like it takes less partial pressure of oxygen to reach that same saturation point. And lastly, once hemoglobin arrives in steeper gradients of partial pressure of oxygen in peripheral tissues, it actually releases its payload quickly and quite well. So you can see in this more steep portion. So hemoglobin in a left shift, at least in the higher oxygen partial pressures, tends to act more like a vacuum of oxygen. It wants to bind to oxygen, it enjoys doing it, and when it binds, it does a really good job of it. But once it plunges into the depths of the lower oxygen partial pressures, below 30, below 20, that's when it really begins to dump all of its oxygen molecules at once. A great example of this actually is this fetal hemoglobin that I spoke about a little while ago. So fetal hemoglobin is a molecularly similar version of hemoglobin found in adults called hemoglobin A. And in fetal hemoglobin, this is characterized by having a higher affinity to oxygen, just like a left shift. So fetal hemoglobin is made by the newborn's bone marrow, and it's present in the child for the first 9 or so months after birth before it shifts to adult hemoglobin production. So the purpose of having a higher affinity to oxygen means to facilitate higher oxygenation in the growing baby. So in summary, a right shift, it's harder for oxygen to bond, it's easier for oxygen to be released, and in a left shift is characterized by a very stable bond with high affinity to oxygen. And a great example of this is fetal hemoglobin. Now we're going to talk a lot more about fetal hemoglobin in the hematology unit, but just as a quick example, I thought it would be a good case study to mention. So let's look at some of the reasons behind this right shift. So right shift is characterized with low affinity, a higher partial pressure of carbon dioxide, and low pH or an increased acidity. This really promotes oxygen release. So how does it do it? So active tissues have increased carbon dioxide, which increases, really through the law of mass action, increase in hydrogen ions. These very ions, they stabilize portions of the deoxyhemoglobin, which promotes oxygen release. When there is higher temperature, this tends to denature the hemoglobin and weaken that hemoglobin molecule or that hemoglobin-oxygen bond. This loosens the bond, and overall it's going to need more dissolved plasma, or oxygen in dissolved plasma, or the PO2, to achieve that same saturation. So carbaminohemoglobin is the binding of carbon dioxide to the hemoglobin molecule itself. And I like to think of carbon dioxide as kind of hitchhiking on the hemoglobin molecule. And we'll see that this is one of about 3 or 4 different ways that carbon dioxide is transported throughout the blood. And this makes up probably about 5% of the total CO2 transport in the blood plasma. But how does this possibly relate to the hemoglobin dissociation curve? Well, with the more amount of this carbaminohemoglobin, it reduces the affinity, or I should say it reduces the affinity and it releases more oxygen into the tissues. So the higher the CO2 that we see in the peripheral blood, we see that there is more of this molecule that is formed, which reduces the affinity. Great, that's number 1. Number 2 is 2,3-DPG diphosphoglycerate, also known as BPG or biphosphoglycerate. Don't be confused, they're the same thing. So this is a naturally occurring organophosphate that is found in the RBCs, specifically made during glycolysis. And when the partial pressures of oxygen are low, we find that there is more of this that is made. So there's an increased production during low O2 environments. We would see this in somebody with COPD, somebody with anemia, perhaps somebody with CHF, and this reduces or decreases the hemoglobin oxygen affinity, which allows for O2 unloading, which is a plus. Carbon monoxide is a molecule that loves hemoglobin. It binds to the same binding sites as oxygen, but what makes it different is that its bond to hemoglobin is like having Arnold Schwarzenegger hold on to that bond with a vice grip. Carbon monoxide loves hemoglobin, and it bonds 200 times more readily than oxygen. The final product is called carboxyhemoglobin, abbreviated CO-HB. And this is the thing that can kill people. So carboxyhemoglobin, it binds faster, it has a much more stable bond with the hemoglobin molecule, and it releases slower. So its half-life is about 4 to 6 hours versus milliseconds with oxygen. Carbon monoxide causes a classic left shift on the hemoglobin-oxygen dissociation curve. So remember that the shift indicates that the stability, it indicates the stability of oxygen molecules with hemoglobin. Well, carbon monoxide, when it attaches to hemoglobin, causes a number of molecular changes to the hemoglobin molecule itself. It forcefully clamps on, and for the oxygen molecules that are already populating the hemoglobin molecule, it suddenly and insidiously stabilizes those bonds too. This is what we call a functional anemia. And it means that you have a normal amount of blood cells, but a percentage of them have been rendered useless because they won't release oxygen. So again, these oxygen molecules cannot be released now because of this bond. For treatment, one needs to increase the partial pressure of oxygen, typically in a hyperbaric chamber with supertherapeutic levels of oxygen. What this does is it overwhelms the hemoglobin unit and eventually breaks that bond. So it would look like being surrounded by new molecules of oxygen. Eventually, this would get replaced. Clinically, carbon monoxide, it's colorless, it's odorless, and chronic inhalation by smoking or in urban environments can disable functional hemoglobin from your blood. About 5 to 8% of arterial blood of populations in urban areas that smoke or are near smoking is composed of carboxyhemoglobin. So an interesting point to know is that people with carbon monoxide poisoning have severe hypoxia. And they have severe hypoxia despite having a normal arterial PO2. Remember that PO2 or PaO2 is dissolved oxygen in the plasma, and alveolar diffusion of oxygen isn't an issue here. It's the reduction of oxygen carrying capacity of hemoglobin, which leads to hypoxemia and eventually death if it gets too severe. This is always a great test question. So this lecture wouldn't be complete without talking about carbon dioxide transport, and we're going to do this fairly quickly. So every minute about 200 to 250 milliliters of carbon dioxide is produced by a resting adult, and honestly this needs to be released as quickly and efficiently as possible to the lungs. It needs to be transported. So carbon dioxide is transported in the blood 3 different ways: dissolved in the plasma, attached to an amino group, and then also as a bicarbonate ion. So going through each one of these quickly, we have already established that carbon dioxide dissolves very readily in solution. It's about 20 times, it dissolves about 20 times more readily than oxygen, and about 5 to 10% of total carbon dioxide is transported in the blood plasma. Another 5 to 10% of this, I like to think of that carbon dioxide hitchhikes on hemoglobin. So what this will do is it will bind to a terminal amino group and get transported peripherally to the alveolar tissues. Lastly, and most importantly, bicarbonate ions are the main mode of transport for carbon dioxide. So we know this in the law of mass action. So carbon dioxide combines with water to make carbonic acid, and this dissociates into bicarbonate ions as well as hydrogen ions as well. So the red blood cell actually has an enzyme called carbonic anhydrase, where it actually allows us to skip this step. This step is actually fairly slow, and what it will do is it will actually move us directly over here, and this is where you can see the carbon located right there. So this is a very fast reaction. So once the hemoglobin and the blood plasma arrive at the alveolar tissue due to partial pressure differences, there is a massive unloading and exchange of gases. These 3 methods is how carbon dioxide is transported in the body. So in summary, we've talked about the partial pressure of gases, we've talked about the factors affecting gas exchange, we've talked about the differences of partial pressures in the tissues, transport of both of the gases, hemoglobin, and its behavior as seen on the oxygen dissociation curve.